suppressing new function. One of melatonin's most important contributions in plants is its ability to enhance stress tolerance. It helps plant cope with various environmental challenges, including extreme temperatures, drought, salinity, and oxidative stress. In addition, melatonin regulates growth, promotes seed germination, delays senescence, and even play a role in photosynthesis. Melatonin has been identified in numerous plant families, including Poaceae, as grasses and cereals, Rosaceae, for example, fruits like apples and cherries, Apiaceae, carrots and parsley, Vitaceae is grape, and Brassicaceae, cabbages and mustard. So on this table presents melatonin in many plants, many species with common name, and of course the place the organ occurs. The right column indicates the concentration or level of melatonin in this plant. Interesting, the concentration of melatonin varies significantly dependent on the species, developmental stages, and environmental conditions. Some plants, such as rice and maize, have been found to certain particularly high melatonin level in the seeds, which suggest a crucial or important role in germination and easily seedlings development. Similarly, medical herbs have been reported to have a variable but often substantial amounts of melatonin, which may contribute to their health-promoting properties. Melatonin was the first found in phototropic marine organisms such as Dinoflagellata, Digulodium polyedrum, before it being detected in macroalgae and evenly in higher plants. In plants, melatonin is widely distributed across various tissue, including roots, leaves, flowers, seeds, and bulbs. However, melatonin level is uniform across species and even within different parts of the same plants. Several factors influence its concentration, including developmental stage of plants and environmental conditions. Of course, in algae there are also melatonin. For example, in Acutodesmus obliquus, my study indicate this fact. Another study has shown that seeds of maize and rice exhibit some of the highest melatonin concentration, ranging from 11 to 2,000 nanogram per dry weight in maize and less in rice. On the other hand, we know that medicine plants have the same similar level of melatonin. The highest concentration was recorded in pistachio kernels, reaching between 227 234 microgram per gram dry weight. We know that this concentration is not static. For example, Arabidopsis thaliana leaves. In Arabidopsis thaliana leaves, melatonin levels range between 80 and 120 nanogram per gram dry weight. But these leaves do not fluctuate significantly through the day. Melatonin production is highly responsive, of course, to environmental stresses. Why melatonin biosynthesis in animals has been well understood for decades. Its pathway in plants is more complete and involves multiple subcellular localization. Melatonin biosynthesis begins with tryptophan, an essential aromatic amino acid. These molecules undergoes a series of enzymatic reactions to eventually form melatonin. The pathway consists of 4 key steps, each catalyzed by specific enzymes. You will see this name of these enzymes. Tryptophan is converted into tryptamine by action of tryptophan decarboxylase. Then, after hydroxylation into serotonin by tryptamine 5-hydroxylase, serotonin is acylated into N-acyl serotonin by serotonin N-acetyl transferase. Finally, N-acetyl serotonin is methylated into melatonin by N-acetyl serotonin orthomethyl transferase or caffeic acid or methyl transferase. Biosynthesis, of course, takes place in different organelles within the plant cell. Under normal growth conditions, melatonin biosynthesis follows 2 primary pathways. The first is from tryptophan, tryptamine, serotonin, N-acetyl serotonin to finally melatonin, and the second from tryptophan via 5-hydroxytryptophan, then serotonin, N-acetyl serotonin, and finally to melatonin. However, when plants experience environmental stress or an increase in cellular serotonin levels, an alternative pathway may be activated from tryptophan via tryptamine, serotonin, 5-methoxytryptamine to melatonin. Regulation of melatonin biosynthesis is tightly controlled by mentioned enzyme activity. The key rate limiting step in this process is conversion of tryptophan to tryptamine, which is catalyzed by tryptophan decarboxylase. When plants need to increase melatonin production, the activity of this enzyme is often enhanced. Of course, additionally, the enzyme serotonin N-acetyl transferase and acetyl serotonin orthomethyl transferase, responsible for final steps of melatonin production, show increased activity under stress conditions. For example, drugs, high salinity or extreme temperature, high and low temperatures. This ensures that plant can rapidly generate melatonin when needed for protection. Unlike many others, phytohormones, melatonin is synthesized in organelles. We know that key enzymes, tryptophan decarboxylase, N-acetyl serotonin orthomethyl transferase, and caffeic acid orthomethyl transferase are located in the cytoplasm. This suggests that the early steps of biosynthesis of melatonin, such as the conversion of tryptophan to serotonin, take place in these organelles. The next enzyme, tryptamine 5-hydroxylase is found only in endoplasmic reticulum, which indicate that the hydroxylation steps converting tryptamine into serotonin is located in this organelle. Then next, of course, the last serotonin and acetyl transferase is found both the chloroplast and mitochondria. The presence of this enzyme, for example, in chloroplast, melatonin may help photosynthesis and protect against oxidative stress. In mitochondria, it may act as an antioxidant to maintain cellular respiration and energy production. In the cytoplasm and endoplasmic reticulum, melatonin is likely involved in signaling and stress responses. This complex subcellular localization also suggests that during plant evolution, melatonin biosynthesis diversified to fulfill multiple physiological functions. The catabolism of melatonin is not fully understood. However, some research suggests that melatonin can be degraded via two distinct pathways. The first is a non-enzymatic degradation. It occurs through reactions with reactive oxygen species and reactive oxygen-nitrogen species. Melatonin can be hydroxylated or deoxygenated by free radicals, leading to the formation of various oxidative metabolites. Another important factors contributing to melatonin breakdown is UV light exposure, which can trigger direct photodegradation, similar to what occurs in animals. And the next pathway is enzymatic degradation. In plants, enzymes catalyze metabolism hydroxylation, leading to the formation of hydroxylated derivatives such as 2-hydroxymelatonin, for example, 2-hydroxymelatonin. These hydroxylation reactions are thought to be similar to those found in animals and other eukaryotes, indicated a conserved function in melatonin metabolism across different life forms in different organisms, plants or animals. The primary melatonin levels. I think that cherry fruits is rich in melatonin. The content is high in diffuse mulberry leaves and is higher in juvenile leaves than in older leaves. Higher levels of melatonin is often observed in seeds, wherein it can elevate the damage caused by oxidative stress. We know that melatonin production in plants is highly responsive to environmental stresses. Both high and low temperatures trigger melatonin synthesis as a defense mechanism. In Arabidopsis thaliana, cold stress significantly increase melatonin levels, helping protect plant from oxidative damage and chilling stress. Similarly, the heat stress in rice seedlings has been shown to enhance melatonin biosynthesis by activating key biosynthetic enzymes. On the other hand, high salt concentration increase melatonin levels in many plants, particularly in the roots and cotyledons. Sunflower seedlings which were exposed to salt stress shown an upregulation of 5-hydroxytryptamines, this means serotonin and melatonin, which is linked to increased activity of melatonin synthesizing enzyme hydroxyindole orthomethyl transferase. In another stress, in drought conditions, melatonin function as a protective molecules, reducing water loss and maintaining cellular integrity and enhancing stress tolerance. Of course, melatonin does not act alone. Instead, it interacts with various classical phytohormones, forming a complex of signaling network that regulates plant growth, stress responses, and development. Melatonin and abscisic acid share overlapping functions, particularly in drought tolerance and stomatal regulation. Under drought stress, melatonin enhances abscisic acid biosynthesis, leading to stomatal closure, reducing water loss, and increased drought resistance in this plant. However, melatonin also acts as an ABA antagonist under normal conditions, preventing excessive abscisic acid accumulation that could hinder growth. Other phytohormones, auxin and melatonin, both influence root development and plant growth. Melatonin promotes lateral root formation similarly to auxin by activating auxin-related signaling pathways. On the other hand, in auxin-deficient plant, melatonin supplementation can partially compensate for auxin loss, demonstrating the functional overlap. Another phytohormones, cytokinins, well known for delaying leaf senescence, but melatonin also inhibited antisenescence properties, and in combination in cytokinins delay aging and improves chlorophyll retention. These interactions makes melatonin an important candidate for improving post-harvest shelf life in crops. Salicylic acid work with melatonin synergistically to enhance disease resistance, particularly against bacterial and fungal infections. When a plant is exposed to pathogen, melatonin can boost salicylic acid levels, activating immune response and reducing oxidative damage. In another interactions, melatonin can interact with osmotic acid pathways to enhance wounding healing and secondary metabolite production, strengthening the plant defense mechanism. Understanding how melatonin interacts with these classical phytohormones open new possibilities in agricultural and crop improvement. Of course, by manipulation melatonin levels, we can improve plant stress resistance, enhance crop productivity and post-harvest quality, and the last develop plants can better withstand extreme environmental conditions. How melatonin regulates key physiological processes such as flowering, fruit ripening, and photosynthesis. synthesis. As melatonin, of course, is a multifunctional molecules that regulates various biological processes. One of the best known function of melatonin is its strong antioxidant capacity. It neutralizes harmful reactive oxygen species, preventing oxidative stress that can damage cellular components such as DNA, protein, and lipids. We know after lectures, many for us said about oxygen stress and the catalase, peroxidase, and other antioxidant, non-antioxidant. Melatonin plays an active role in flower induction and reproductive development. It helps regulate photoperiodic resistance, ensuring that flowering occurs at the optimal time. It's good for agriculture, of course. In fruit-bearing plants, melatonin is involved in ripening processes, particularly through its interaction of ethylene and auxin. For example, in tomatoes, melatonin was found to promote ethylene biosynthesis, accelerating fruit ripening while also enhancing fruit quality and antioxidant properties. Melatonin also improved photosynthetic efficiency by protecting chloroplast from oxidative damage. Melatonin delays chlorophyll degradation, maintaining the higher photosynthetic rate and improving plant productivity. In crops such as wheat or rice, melatonin treatment has shown to increase biomass accumulation and enhance overall yield. Melatonin plays, oh, maybe other, oh, it's nice. At cellular level, melatonin protects chloroplast and reduces the red chlorophyll we know. Increased stomatal conducts, optimizing gas exchange and water regulation. Why is it important? Understanding how melatonin regulates fruit ripening, stress tolerance, and photosynthesis can lead to practical application in post-harvest technology and crop management. By applying melatonin treatment, we can extend the fruit shelf life and quality after harvest, enhance fruit sweetness and weight, improving market value, boost photosynthetic performance, leading to healthier plants and increase crop yields. Melatonin also participates in regulation biotic and abiotic stresses. The combination of hydroxyperoxide and nitric oxide, mitogen activated protein kinase, cascade, and oxidative signal inducible kinases are responsible for melatonin triggered innate immunity signal transduction. During this process, the gene encoding the salicylic acid biosynthesis enzyme isohorizmate synthase is upregulating, leading to increased salicylic acid levels. In summary, the protective properties of melatonin are: melatonin acts as a free radical scavenger, enhances activity accumulation of antioxidant enzymes, enhances the redox status of cell via regulation of glutathione synthesis and oxidation, ensures the protection of antioxidant enzyme from oxidative destruction. Melatonin reduces free radical formation, electron leakage by enhancing the efficiency of the mitochondrial electron transport chain. This graph summarizes the function of melatonin in plants. I hope we have a printed version. You may stay longer. The next group is strigolactones, a class of phytohormones that play, as I said earlier, a key role in plant communication, development, and stress adaptation. Unlike traditional phytohormones, strigolactones serve two major functions. The first, they regulate plant growth and architectures, much like auxin and cytokinins. But what makes them truly unique in the second role? The second role is chemical signals in soil, mediating interactions between plant and microbes. Strigolactones are carotenoid derivatives compounds naturally occurring in a wide variety of plants. Of course, not in algae. Not in algae. They were originally identified as root exudates, meaning they are released from plant root and into the soil. Strigolactones were first identified in 1966 when researchers studying cotton plants, similar, of course, to India, found certain compounds in the roots exudate triggered the germination of parasitic plants like Striga and Oronbacher. Initially, these compounds were considered mere chemicals, substance released by plants and that influence neighboring organisms. However, decades of research revealed that they are actually a new class of plant hormones with far-reaching effects on plant physiology and biochemistry. The first naturally occurring strigolactone to be identified was strigol, from which the entire group gets its name. Since then, the scientists have discovered over 45 different strigolactones in various plant species. Some of the most well-known include Oronbacher, sargolactone, solanacol, and sargomol. Strigolactones are carotenoid derived lactones, composed of three primary components. The first is ABA 3 cyclic core, a fuse ring system responsible for biological activity. And the next, the butanolide D ring, is has a crucial functional group that interacts with strigolactone receptors. And then connecting a known bridge, the structure likes the ABC core to the D ring and contributes to chemical stability. The D ring is particularly important because it serves as a bioactive signaling unit in plants and the rhizosphere. Some chemical structures, of course, strigolactone divided into canonical and non-canonical strigolactone, strigol, Oronbacher, heliolactone, zalolactone, avenol. More compounds are in the last book, Professor Shamsul, Professor Bahri, and me. Edited, of course, not ours, book edited. We'll see on the first day. Strigolactones can be categorized into two main groups based on the structural differences. The first is canonical strigolactones. These compounds have a characteristic tricycling lactone ABC structure connected to the butanolide D ring. Example includes on these slides. Canonically, strigolactone are primarily involved in shoot branching inhibition and mycorrhizal signaling. Non-canonical is the second type of strigolactones. These molecules lack the typical tricyclic ABC core, but still contain the bioactive D ring. They include simplified and modified derivatives from found in specific plant species. Non-canonical strigolactones are more diverse and often play a specific role in rhizosphere interactions rather than shoot regulation. Those molecules lack the typical. We know about it. Some examples are present. In 2008, strigolactone were categorized for the first time as a new class of plant hormones in recognition of their various role in controlling above ground plant architectures, for example, by inhibiting bud outgrowth and in underground communication with adjacent organism. Strigolactones were originally considered to belong to the sesquiterpene lactones, a widespread family of compounds formed in a cytosolic compound farnesyl diphosphate. Biosynthesis begins in the plastid, where carotenoids are broken down by carotenoid cleavage dioxygenases. The key enzyme in this process is dwarf 27. In abbreviation is D27, which convert all trans beta carotene into 9,6 beta carotene. It is followed by two critical cleavage reactions catalyzed by CCD7 and CCD8, resulting in formation of carotenoid-derived precursors, which are further modified to produce active strigolactone. The enzyme more axillary growth, as abbreviated as MAX1, cytochrome P450 monoxygenase, more known as MAX catalyze, a sequence of oxidation reactions converting carlactone, the first precursors of strigolactones, into carlactonic acid, and then is processed into methyl carlactonate, precursors the various strigolactones. Strigolactones serve dual function as both endogenous and exogenous signaling molecules. With the rhizosphere, these compounds are responsible for arranging the associated between plant roots and arbuscular mycorrhizal fungi, particularly in nutrient-deficient environment. They also play an important role in germination of parasitic plants, Striga and Orobanche. Strigolactones also plays other important roles in the root system. enhancing the length of primary roots and root hairs, encouraging growth and rise crown roots, and suppressing the creation of adventitious roots in, for example, tomato, Solanum lycopersicum, Arabidopsis thaliana, and Pisum sativum. Strigolactone play a dual role in underground signaling. As I said earlier, influencing both parasitic seed germination and the growth of beneficial mycorrhizal fungi. Their function in the rhizosphere determines whether a plant will form a mutualistic relationship or face a parasitic threat. In the left side, yeah. In the left side, when a host plant exudes environmental into the soil, these compounds act as a germination signal for dormant parasitic plant seed in Striga or on Bache. Once activated, the parasitic seed germinates and begin searching for a host root. If the seed radical successfully reaches the host root, it established a parasitic interaction attaching itself to the host vascular system. It leads to nutrient depletion in the host plants, reducing its growth, weakening its structure, and in certain cases causing crop failure. This interaction is especially problematic in cereal crops such as maize, sorghum, and rice, where parasitic weeds cause significant yield losses worldwide. Right side of this pictures present that strigolactones play a positive role in plant health by mediating beneficial symbiotic relationships with arbuscular mycorrhizal fungi. When a plant releases strigolactone into the soil, they are sensed by fungal hyphae, which respond by directing their growth toward the plant's root system. Once contact is established, the plant and fungus exchange essential nutrients. The plant provides sugars and as an energy source to the fungi. In return, the fungi supply nitrogen, phosphorus, or other nutrients to the plant, enhancing growth and stress resilience. This mutualistic relationship is particularly important in nutrient-poor soil, as it significantly improves plant nutrient uptake and overall fitness. Strigolactone influences key development and traits, including seed germination, early seedlings development, ensuring optimal establishment, internode elongation, affecting plant height and canopy structure, leaf morphology and senescence, helping plants adapt to environmental conditions, should gravitropism, enabling plants to adjust their growth direction in response to gravity, stem morphology, influencing plant architecture and biomass distribution. One of the most striking effects of strigolactones is their role in accelerating leaf senescence. Strigolactone interact with abscisic acid, another key phytohormone involved in stress signaling and draw response. draw response. We know that strigolactone play an important role how plants respond to osmotic stresses and drought conditions. Of course, they interact with abscisic acid, regulate root-to-root signaling, they influence stomatal closure, all of which contribute to water conservation and stress adaptation. In normal conditions, strigolactone are produced in root and transported upward to the shoots where they suppress the transcription of strigolactone biosynthesis genes, ensuring that strigolactone levels remain balanced. However, during drought or osmotic stresses, the synthesis of strigolactone in roots is inhibited, reduces the upward flow of strigolactone molecules. As a result, strigolactone biosynthesis genes in shoot became active, more active again, leading to an increase in strigolactone production within a shoot itself. Once produced in a shoot, strigolactone contribute to stomatal regulation, helping plants minimize water loss. Of course, it's summarizing. It means that strigolactone act as a both direct and indirect regulators of stomatal movement, ensuring that plants retain water efficiency during stress conditions. Strigolactones also plays important role in plant immunity. These hormones influence gene expression, interact with key defense-related hormones, and even inhibit pathogen growth, making them important regulators of plant resistance against bacterial or fungal infections. Application of synthetic strigolactones was found to strongly inhibit the formation and growth of phytopathogenic fungi. So strigolactone not only modulate plant immune responses, but also act directly against fungal pathogens, potentially offering new strategies for natural plant protection. Strigolactone may also help plant overcome pathogen-induced stress at the transcription level. They interact with the proteinase inhibitor 2, PIN2 gene, which plays a role in plant wound and pathogen response. Of course, strigolactones contribute to a fine-tuned immune response, ensuring that plant efficiency defends itself without unnecessary energy expenditure. Strigolactones act as a mobile signaling molecules that are synthesized mainly in roots and transported to the target tissues, where they influence various physiological processes. In this target cells, strigolactones are per se by specific receptor protein called dwarf L14. This receptor belongs to the alpha-beta hydroxylase superfamily and plays a dual role. It not only binds strigolactone but also hydrolyzes them, triggering the signaling cascade. Once strigolactones are detected by D14, the receptor interacts with MAX2, we know the full name, and F box protein that plays a key role in signaling transduction. MAX2 is a part of ubiquin ligase complex, which function as a molecular switch that regulates plant growth responses. Under normal conditions, a receptor protein known as dwarf 53 inhibits the downstream effects of strigolactone, preventing changes in gene expression. However, when strigolactones are present, D14 MAX2 complex mark D53 for degradation, effectively removing its inhibitory effect. D53 is degraded, genes responsible for strigolactone response became active. It leads to several physiological changes, such as the inhibition of shoot branching, modification of root growth pattern, and improved adaptation to environmental stresses. In a sense, the degradation of D53 allows strigolactone to exert the full effect, ensuring thus the plants can adjust the growth and development based on external or internal cues. Strigolactone and maybe I told interactions. Yeah, it's probably, yes, it's the most probably. This is the end. Thank you.